Now that we have calculated the declustering from multiple sources, we can compare all the declustered distributions. So I'm going to run this cell and then I'm going to explain what we're doing. So we're initializing a figure with 4 by 4 array. We are iterating over all the domains that are stored as the softcomps dictionary. So with that, we have access to the domain name and the composites of that domain. We build univariate statistics for each one of the weights: the nearest neighbor, the inverse distance, and the cell declustering. And also we generate a unweighted distribution when we specify the weight to be none. That is saved on this unistats, which is just a list of all the univariate statistics. We specify the names for each one of these statistics so that they match the order that we specified for the weights. So the first one is none, so that means this is clustered, not declustered. Second one is the nearest neighbor. Third one is inverse distance. Then the fourth is the cell declustering. Now that we have created multiple univariate statistics, we initialize a unique compare object that is used to compare multiple univariate statistics. We initialize it with all the individual univariate statistics and the names that matches that list. Next, we use the univariate comparison to plot a CDF plot with overlap. So that just means that we are plotting all the CDFs on the same figure axis. So when we are iterating over the domain, we set the title to match that domain. So that for the outside domain, these are all the distributions. We have the clustered, we have the cell, the clustering, inverse distance, and nearest neighbor. Because inverse distance and nearest neighbor are using the grid cells, they are more similar to each other than they are to the cell declustering, which is not using the cells of the given domain. We can also see the inside distributions, and a similar behavior is noted. On the last cell line of the cell, we are also adding table plots to the axis. So these table plots, they will match the CDFs above, just providing the summary statistics. So we can see how the average is changing depending on the technique that we use. The average is the same for the outside domain when we compare nearest neighbor and inverse distance, but it's quite different from cell declustering. Because the extent of the domains are well defined by the grid, the representativeness of each sample relative to their domains, it's more well captured when we use nearest neighbor or inverse distance compared to cell declustering. So we do recommend to use one of the two when we do have a grid and the domains defined. We see a similar behavior when looking at the inside domain. The averages are similar for nearest neighbor and inverse distance, and quite different for cell declustering for the same reasons. To finalize the declustering session, we need to choose which weights we are going to use moving forward throughout our workflow. And we are choosing here the inverse distance weights. The choice of inverse distance is because inverse distance is typically more robust in terms of not assigning extreme weights to samples at boundaries of domains, and also is more robust in terms of not assigning zero weight for given composites if they are not the closest to any blocks. So inverse distance is typically more robust than nearest neighbor, and we have selected inverse distance here.